interruption. Simultaneously, another session is going on at the second floor.
Good morning, Chair, ladies and gentlemen. First, I'd like to show my thanks to organizers for the kind invitation. I'm very pleased to be here to meet so much new friends. I'm from the city of Chongqing, China. It's Beijing, it's Shanghai, it's Tibet. Here's Chongqing. Chongqing sits on the upstream of the Changjiang River and almost at the central part of China. From the view of population, Chongqing may be the largest city in the world. There are more than 30 million people, so it's almost equivalent to the total population of Canada. So Chongqing is one of the 4 municipalities directly under the central government of China. It's also the capital during World War II. In that university, we have a medical university called the Third Medical University. In that university, I have an institute for immunology. That is also the National Engineering Lab for Hepatitis Therapeutics and the National Center for Immunoproducts Research and Development and the National Base for Bioindustry. In our institute, we have 3 units. The first unit we called clinical immunology centers. We have 3 at different 3 hospitals. The second unit is for basic research of immunology, and the third one is for R&D. So usually we start our research from clinic and then go to basic immunology and R&D. So it's a typical B2B study. Our research work focuses on persistent infections and autoimmune disease. My talk today is briefly to take your story about HBV infection. You know, HBV infection is very popular in the world. It is estimated by WHO more than 30% of the whole population had infected with the virus. And the non-response rate to the preventive vaccine is around 5 to 15%. The persistent infection of the virus could aggregate to leukophilia, cirrhosis, and carcinoma at a rate around 20 to 30%. HBV infection is especially important in China, for there is a very high percent rate of carriers, around 8%. So we call it the disease of the nation. HBV infection could induce acute hepatitis, carriers, chronic hepatitis, ACLF, cirrhosis, and carcinoma. But most part of the infected people recovered with or without liver damage. As the virus is a stealth virus, so the different outcomes was determined by immunity. This is the rationale to develop the therapeutic vaccine for HBV infection. Currently, therapies therapeutics mainly are interference and antiviral drugs. In terms of safety and efficacy, they are not satisfactory in clinic. So there is a very clear medical need in clinic for new therapies. Therapeutic vaccines are primary means to treat disease. But so far, by using natural antigen, such as different forms of preventive vaccines and DM vaccine, could not get good results in clinic. So we reached a new strategy we called Mimogen. From view of strategy, so far the pre-exposure vaccines, or rather to see the preventive vaccine to take the genus strategy. That is to mimic the immune response from natural infection. That is very successful in smallpox, polio, and measles, and so on. But for persistent infections, tumor, autoimmune disease, and cognitive disease, The natural antigen already exists. Heterologous immunity already exists, but disease still progress. So how to make a vaccine for this situation? For HBV infection, we proposed two-wave theory. The first wave is induced by the virus. If the first wave is strong enough, the virus cleared and the patient recovered. But if not, the second wave will arise. So during the second wave, the CTL exhausted and the endogenous danger signal released. So the disease deteriorates. So we hope by using Mimogen, we can mimic the first wave or better than the first wave for therapeutic use. As you know, immune response is based on antigen, or rather to say based on epitopes. So different epitopes induce different immune response. For therapeutic use, the pathologic immune response should be deleted, and the protective immune response should be enhanced. So based on good epitope, natural epitopes, or reconstituted epitopes, we can build a new antigen we call Mimogen. It's a word created by me, and it means it's not a natural antigen. It is also not a mixture of epitopes. It is one molecule containing different epitopes. We hope this kind of Mimogen could remove the immune response for therapeutic use. The conventional target for HBV therapeutic vaccine to some extent is clear. As in HBV transgenic mice or acute infection of HBV infection in human, the CTLs can inhibit HBV gene expression and replication without killing hepatitis. But we don't know how about CTLs in human chronic HBV infection. To answer the question, different cohorts of patients was employed to assess the function of tri-epitope-specific CTL in acute chronic viral hepatitis B. Tetramer and pentamer were employed to determine the CTL frequency number. And two models for human CD8 T cell differentiation were employed to define the differential status and functional status. One is based on the expression of SSR7, another is based on CD27-38, and CD45I and RO. Generally, we found in chronic infection of HBV, the CTL function is of heterogeneity. More than 90% of HBV-specific CTL are at the intermediate or late stage. They could not express no gamma, granzyme B, or perforin. Most part of the perforin granzyme positive cells are bystanders. So finally, we define the several epitopes with therapeutic potential. Recently, we moved target to the CD8 T cell repertoire. In brief, for each individual patient, the CD8 repertoire could be divided into four parts. A is HBV infection specific. B is shared by HBV patients and health donors. For A is related to the E antigen antibody seroconversion. If A is strong enough, seroconversion will be happened, and it's not related to liver damage. So by using A, we can predict the antigen antibody seroconversion with a very high accuracy. Just in opposite for B is related to liver damage, but the negative related to seroconversion. Further, if we use AB ratio as a new indicator, the prediction accuracy will be more powerful to predict the gene antibody seroconversion with a more high accuracy. So in summary, in chronic infection of HBV, most of the CTLs are at the intermediate or late stage. They could not express gamma, granzyme, or perforin. The protective repertoire exhausted. That is related to virus load. But to liver damage, it is related to harmful repertoire and bystander cells. So how to design and screen Mimogen? We set up a new technology called epitope-based vaccine design, EBVD. First, we set up the epitope and the subtype database for viral antigens. And based on the database, we established several epitope prediction methods and a cytokine system for epitope experiments. Based on the cytokine system and the experimental system, we got the epitome of HBV with a different background of the Chinese people. During our study, we found one kind of epitope is quite special. We call it non-lytic epitope. It means it can downregulate the virus load without liver damage. I think it's the ideal component of the therapeutic vaccine. Theoretically, the epitope could improve the binding to MHC and TCR. By improving the binding to MHC, we could enhance the T-cell response. And by improving binding to TCR, we could activate low and high avidity T cells and tune the repertoire, even accurate the cross-reactive T cells. Even we can generate T-cell antagonists for negative vaccine. Here example to show you by improvement of binding MHC enhanced antigenicity, avidity, and affinity. This example to show you improvement of binding TCR circumvents the tolerance by eliciting different T-cell repertoire with higher avidity. Another key is the delivery system. We try many, but finally we found three kinds of delivery system could induce CTL response in vivo. One is for particulation. The second is building adjuvants. The third is target to the MS class 1 restricted person pathway. For easy approval by SFDA, we take liposome as the delivery system. The size of the liposome is around 80 to 100 nanometers in diameter. And the encapsulation efficacy is very high. It can the Mimogen could be reduced at different conditions and different times. And generally, the half-life time within the body about around 5 days. So, based on the finding I mentioned above, we got the Mimogen we called epsilon PA44 with the building adjuvant and the liposome delivery. We conduct two clinical trials, randomized open-label dose escalation trial and a randomized double-blind placebo-controlled trial. Briefly, the baseline between health donors and patients are very similar. A vigorous CTL response was evoked in health donors with 3 injections with an interval of 3 weeks with a minimum of 300 micrograms. Similarly, in patients, we saw 4 injections with a 6 weeks interval and minimum dosage at 600 micrograms could induce vigorous CTL response. The human response was also invoked both in health donors and patients. And the Mimogen could drive the CTL status from pulse status to functional status. If the CTL are arise, the virus load will downregulate and finally seroconversion happen. To our surprise, at the same time, the Treg was downregulated. The liposome changes the repertoire in a coordinated way, while Mimogen changes structural characteristics of the repertoire. And the protective repertoire was induced and sustained by the Mimogen vaccine for more than 17 weeks. So generally, the vaccine is safe. For long-term follow-up, we found in 7 of 9 patients, HBV load decreased more than 2 logs within 132 weeks. 6 of 9 serum seroconversion was happened within 132 weeks, and no relapse was happened to 252 weeks. The final question is how this kind of particle vaccine could induce vigorous CTL response in vivo. As the antigen process is processing is a process of membrane fusion and vesicle transportation. So we focus on rabbit family. Altogether, we construct 171 lentivirus based on SRNs against the 57 members at that point. On the other hand, we established stable antigen cross-presentation as in vitro. In brief, we found altogether 15 rabbit family members are related to the cross-presentation and can be classified into 3 groups. One group is related to fungicide, and another related to the transportation from fungicide to lead enzyme. And last group is related to the cytosis vesicles. Finally, we found this kind of particle vaccine could be engulfed by APC, and then with the help of RAB32, it can transport on the lead enzyme, and with the help of RAB3B, RAB3C, the positive vesicles, it transports onto the cell surface, and this kind of MHC peptide complex could be recognized by T cells, and the T cell then activated. I would like to show my thanks to my colleagues, and thank you all for your attention.
Thank you, Professor Wu. Are there any questions? I have one. What do you think is the mechanism? You mentioned that you think the T-cell mediated protection would not induce liver damage. How do you imagine the CTLs are going to clear virus without damaging some of the liver cells? So
we found the HBV-specific CTL could downregulate the virus load without liver damage.
So they're not going to act as a lytic. They're going to downregulate virus expression? Is that what I heard you say?
Let's see the transgenic mice. We found maybe by a soluble mechanism. We just want to find out the soluble molecules. Conventionally, the CTL could be used phospholigand and peripheral enzyme to induce the apoptosis of the target cells. But in HBV infection, we found maybe we have another way. They call non-lytic epitopes or non-lytic eradication of the virus. So we want to search for the molecule, maybe a soluble molecule, I think.
Okay, thank you. Nick?
So if I understand you correctly, the patients are getting control of the virus but not eradication of the virus. And that study you presented was done a number of years ago. So is this now in broader use? What's the strategy? Are you seeking approval of the vaccine, or just where? Can you explain what stage it's at?
Yeah. The heterologous immunity was happened in the chronic infection of the virus. So the different status of the immunity determine different status of the disease. So the remaining immunity especially the CTL response, is essential for control the virus load, even high or low. So how to mimic the first wave of immune response? I mean, the automatically recovered by the CTL response. That's my strategy.
Hi, thank you for your presentation. I saw that one of your epitopes you're basing your mimotopes was on the C1827 core peptide, which is an HLA-A2 restricted, and that's a very Western-dominated HLA phenotype. Have you shown cross-reactivity to be loaded on more Asian populations like A11, A07?
Yeah. The vaccine is only for A2 restricted people, I think. Yeah, thank you.
Yes.
So, in your recovery of your CD8 responses, are you saying that it's a de novo CTL response, or is it recovery of the exhausted CTLs?
So far, what we observed is the exhausted CTL response could be re-evoked and sustained. And sustained if I think it's needed. If you walked and if done rapidly, the virus load or the seroconversion will never happen. So it should be walked and sustained for more than 70 weeks. The seroconversion will gradually happen from my observation. Yeah.
Okay, thank you very much, Dr. Wu.